Welcome back to the Molpigs podcast. Today our guest is William Poole. Also with me today are Boya.
Hi.
Georgios.
Hi.
And I'm also Will. William Poole received his B.Sc from Brown University in biological physics and is scheduled to complete his PhD in Computation and Neural Systems from Caltech shortly, co-advised by Erik Winfree and Richard Murray. His interests involve developing mathematical and computational tools to understand how cells think and how to program cells. His work has spanned theory, computation, and experiment. William, hi.
Hello.
So, your work has kind of spanned quite a few different topics. Could you maybe give us a brief overview of some of the sorts of things you've been doing?
I guess I want to know everything, so I try to do everything, which is not always the best idea, but it certainly keeps me occupied. I guess my background is fundamentally in physics and programming, computer science, and I've tried to apply that in various ways to understand and engineer biological systems. This has included both bioinformatics types approaches and also more recently trying to use machine learning to engineer and reverse engineer biochemical networks, in particular by exploring mathematical similarities between biochemical models and machine learning models so we can try to exploit those similarities to do cool things.
So this is your kind of chemical Boltzmann machine work, right? Is that what you're talking about with the machine learning work?
That is one example. In that work, I related how certain implementations of machine learning algorithms could be built in theory by chemical systems. But I think more generally, my work is very inspired by what's been successful in machine learning and how we can apply that to synthetic and systems biology. So to elaborate a little bit, if you look at, say, 30 years ago, machine learning wasn't getting very far. What really happened was that we had large data sets, faster computers, and really good libraries of software that allowed machine learning engineers to explore topologies of different networks and actually test and understand sort of empirically as a science what things worked for machine learning and what things didn't. And we need to do something similar with biochemical systems to both understand them and engineer them. And that requires a mixture of theoretical tools and software tools, as well as large data sets.
And I think you've been working a lot on the software tools side of things, working on software such as Biocompiler and Vivarium. Sort of if you could just quickly tell us what those are.
Yeah, so Biocompiler is inspired by these machine learning tools like PyTorch, and it allows you to basically compile or build large, complicated chemical models of different biochemical systems without, really having to get into the nitty-gritty specification of every single interaction. So it says things like, I have a gene, and that gene can be transcribed and then translated. And if I tell Biocompiler that information, it will work out what all of the different chemical reactions should be. And it does this in, I think, a kind of clever, different way. At least I think it's clever because I came up with it, which is to think about models in sort of three terms. We have what's in the model, the components. We have the context of the model, which we call a mixture. And we have mechanisms, which are sort of the microscopic descriptions of how a given process occurs. And by mixing and matching components, mechanisms, and mixture context, we can compile a huge number of models that are very diverse or multiple variations of the same model that maybe have different assumptions in them. And I think you also asked about Vivarium. Vivarium is a similar idea, but instead of compiling models, it's now more of a simulator, and it gives you the ability to link different simulators. So, I've mentioned chemical reaction networks many times here because I work on them a lot. They're just one of the many ways we might simulate biochemical systems. Maybe you need mechanistic, mechanical simulations to understand how cell membranes work, or, some sort of mechanical simulation of a DNA polymer. Vivarium lets you couple different simulators together in a seamless way, and then simulates them and takes into account the scheduling and the updating, etc.
So it sounds like, you've spoken a lot about chemical reaction networks, and obviously that's one of the main focuses of your research, but also you're building tools, you're building tools for synthetic biology, and I believe you also go into the wet lab every now and then, and do some synthetic biology as well. So it seems like you're kind of straddling the interface between molecular programming and synthetic and systems biology. Like, would you agree with that?
100%. I try to sit right in the middle. I think that molecular programming gives you tools for abstraction, which are necessary, I think, if we want to understand biochemical systems holistically. I think that synthetic biology lets you engineer things, and it's often quoted that you don't understand something if you can't engineer them. I think Feynman said that. I am a believer in that notion. But systems biology is really, that's the context of where synthetic biology lives or where molecular programming is likely to be deployed. So if we don't understand the living organisms that we're working in and around, we're not going to be able to engineer anything that can work in those environments. So you can't detangle these three areas in my mind.
So you can't detangle them, but do you think you can draw a boundary as to where someone's research stops being molecular programming and starts being synthetic or systems biology?
I mean, lots of people draw boundaries. I think that the best science happens in the middle.
So have you, what kind of applications have arisen from your tooling so far? So Biocompiler and Vivarium, have they started to be used by others in any way?
Yeah, many people have used Biocompiler, mostly sort of in the greater Caltech community, to model different biochemical circuits and various things ranging from, E. coli in the gut to cell extracts. I know there was also some systems level biology models that came out of one of our classes that someone used around extracellular membrane proteins or something like that. It was a little outside of the scope of things I know a lot about. We are actively, wanting to get more users and have invested a lot of time in building educational materials so that people can learn to use this software tool and deploy it. But, that takes time. In terms of Vivarium, I've only recently been involved in Vivarium for the past, 6 months. It's a much newer tool. It's only maybe a year old. It's being extensively used by the Covert Lab in Stanford, which does whole cell modeling. So I think they have released already a number of papers using Vivarium to model E. coli at a systems level, or even E. coli colonies. And I think that Vivarium has a lot more potential than just modeling whole cells. It could be used to model whole organisms, or also to sort of link systems level biochemical models to individual molecular biophysical models.
I just want to point out that I have been using Biocompiler on and off for my own research since you gave a tutorial for Build-A-Cell back in late 2020. And I guess what we want to ask is, how did you actually get involved in Build-A-Cell? How did you get involved and get into a position where you were giving a whole series of lectures about your tool?
Well, that wasn't just about my tool. There were a number of tools in those lecture series. Build-A-Cell started I think it was sort of a thought child of Drew Endy and Richard Murray and a couple other people. And the first meeting was at Caltech. And, for me, it was pretty easy to just take my coffee and go into the room. And it's like, oh, these guys are kind of trying to do something cool, which is, building a cell from scratch. And it was a whole bunch of people bringing different perspectives to that. It was really cool meeting the guys from the Craig Venter Institute who are doing all this minimal cell work. And these are some of the people who pioneered understanding how a lot of biochemical machinery worked 30 years ago and are now, taking that deep experience to systems biology in a way that I think is phenomenal. There's also newer people in Build-A-Cell doing really cool things. Kate Adamala is doing fantastic work in building vesicles that interact. And I think if you, as a systems biologist, it's very, very hard to always know that you're right. There's too much complexity. But when you can start sort of engineering the systems you're trying to understand, say by using cell extracts or protocells, it allows you to validate the types of tools and techniques you want to apply to systems biology in a situation where you can actually really know that you're right. And I think that's, to me, sort of what's very powerful about Build-A-Cell.
For listeners who aren't aware, we have it interviewed Kate Adamala too, so you can check out more of what William was saying in that episode. So would you say that Biocompiler and Vivarium are kind of complementary tools that are very useful in this kind of approach, because you can, say, use Biocompiler for the more low-level stuff and Vivarium to look at the high-level overview of these systems?
They're different. Vivarium is a simulator, and it's actually not even a simulator, it's a meta-simulator. Vivarium is a series of wrappers that go around many different simulators. We have in the Murray Lab software called Bioscrape, which is our chemical reaction network simulator, that plugs into Vivarium. Biocompiler is not a simulator, it is a model compiler. So it generates the models for you. It's sort of like when you type Python code, that code is interpreted to become something your CPU can run. Biocompiler generates code that a simulator can run, but you don't have to write the code that a simulator needs. You write something much higher level, much simpler. It's a compiler. So they are complementary, but they do very different things.
So I haven't used Biocompiler before, and I know that there are some other software tools that is rule-based, or some other softwares that can simulate stochastic chemical systems. So I wonder, how is the tools that you are building different from other tools?
Biocompiler is unique. As far as I know, there's really nothing quite like it, except for compilers. The inspiration is these sort of machine learning libraries where people say, I want to have, a layer, this topology for a neural network, and it then, compiles that into something that can run on your GPU. These rule-based models are much more like chemical reaction network models. They're sort of ways of simulating things. And the rules give you a way of saying what happens next. That doesn't tell you like, oh, I have some, organism, it's an E. coli, I put this gene into it, tell me all the rules that should happen, right? That's more of where Biocompiler is living, is a much higher level description by having a large built library of validated models. So in systems biology and synthetic biology, many people use models, and there's tons of them published, but they're rarely reused. And Biocompiler tries to sort of re-implement those models so you could reuse stuff that already exists rather than programming it from scratch.
So how do you choose? I imagine there is a library of different models, and how do you pick? How does the software pick which model to use?
You pick. So you tell the software, I want to use this model, and it will then compile using that model. And how do you pick? Modeling is an art as much of a science. And I think part of the inspiration for building this software was so that people could explore different modeling choices more quickly. So in my own research, I'm interested in cell extracts. I think of them as sort of a pseudo-living system. You take E. coli, you lyse them, you separate out all the DNA and lipids and just keep sort of the proteins and cytosol. And then you can put your own DNA back into that system and watch it run for a while. And so when you make these models of cell extract, it's not always clear what the right model is. And I found myself, spending a lot of time playing with, if I have 3 genes that talk and I want to make one change to how I model transcription, I have to make it 3 times. And I was like, it'd be much easier if I could just make that change once. And what if I want to sort of do a combinatoric set of changes between transcription and translation and another process? Now it becomes much more complicated to keep track of all the models. Biocompiler makes that easy. I just say, loop through my 3 transcription mechanisms, loop through my 3 translation mechanisms, loop through my other mechanisms, try them all out. And then I can choose the model that makes the most sense based upon my experimental data, or, the complexity I'm looking for, or the analytic tractability I'm looking for.
How far can you go with Biocompiler to kind of say tweaking these systems? Like when you're talking about making changes at all these different levels, like what is the furthest you can go at this point, or am I misunderstanding?
If you're willing to write your own classes and mechanisms, you can go as far as you want. It's just anything a CRN can represent, Biocompiler can represent. It's just a way of thinking about models as it's a way of factorizing models is really how I think about it, right? You have a CRN is a lot of reactions and species, but you can factor them. There's common motifs that appear over and over again. And so you can factor those as boxes. Those are what we call mechanisms. And you can then replace that box with a different box. And you can then factor sets of mechanisms as sort of components. And And so you could, if you're willing to write your own code, you can take this wherever you want. It's a framework. We have a library that's really focused on E. coli synthetic biology because that's where the people who developed it, what we work in. But there's no reason that's the limit. In fact, I've written grants to try to do this with plant biology, for instance. No one gave me the money, but maybe someday they will.
So it seems like in Biocompiler there is this hierarchy of mixtures, components, mechanisms. The mixtures I'm interested in. First of all, what mixtures are available, and how is the user assured that they are, say, validated or realistic enough? Is my transcription system behaving realistically in this particular mixture?
Well, so the mixtures are sort of models that we have been using for many, many years to understand E. coli at various levels. These are the type of models people like Michael Elowitz use when he models his circuits. They're the type of models that have been published in textbooks and in seminal papers. Are they correct? No model's correct. All models are wrong. Are they good enough? Often. The real question, I think, to understand if a model is good enough is, does it explain the experimental data I observe? And we actually have other software in Bioscrape that does parameter inference. It's really sort of more of a link between Bioscrape, which is our CRN simulator, and this thing called MC, which is a Bayesian parameter inference package. And the way I think about this is, if I really want to know if one model is better than the other, I should try to infer all of my unknown parameters from my data. And if one of the model both fits the data better, but also has highly inferable parameters, that's the best model. That's sort of the best I can do. On the other hand, if another model doesn't fit very well, even if I can infer the parameters, that's probably not very good. Or if a model doesn't fit or if a model fits very well, but the parameters are like way overfit, that's probably also not very good. So you can't think of a model, by itself as right or wrong. It's really a model exists in a context, and that context is not only sort of the biophysical context, but it's also the context of the data you're using to validate that model.
So you work quite a lot in theory, computation, and experiment in the wet lab. Which would you say comes easier to you? And if you had to pick one for the rest of your career, which would it be?
I'm not going to pick one for the rest of my career. I'm going to continue to do multiple things. Look, I'm a very good programmer, but programming all the time without strong theoretical skills is a great way to write bad code. I think building tools without understanding what it's like to work in the wet lab and without understanding the needs of actual bioengineers or systems biologists is also, a dead end. You really need to work closely with the people who are going to use your tools. You can't just build, tools that aren't helpful. If I built Thor's hammer and then realized no one could lift it, that wouldn't be useful for anyone to hammer in a nail, which is what people really want to do. Theory, I think, is super fun, but theory has the same sort of problems that, if I do esoteric, ivory tower theory that doesn't even relate to the types of measurements people are taking in the lab, my theory is not very useful. Similarly, if my theory is too complicated that no one else can manage to handle or understand it, it's also perhaps not very useful. So I really want to think about theory in terms of, what's theory that is practical and applied theory. Math is very, very powerful. Physics is very, very powerful. But when you try to apply these ideas to complex systems, often the simpler ideas are the ones that really stick, because when you start scaling them and mixing and matching them in a combinatoric number of ways, you can't handle anything too complicated as your underlying piece. You have to use simpler pieces because the complexity just comes from the scale.
So tell us a little bit about the theory work that you have working on.
Well, so I'm really excited about a new project. This is a follow-up of the chemical Boltzmann machine work. The working title of the paper is Detailed Balanced Chemical Reaction Networks are Generalized Boltzmann Machines, so it sounds quite similar. But what we've really done is we've combined ideas from information theory, statistical physics, machine learning, chemical reaction network theory to show that a broad class of biophysical systems, detailed balanced chemical reaction networks, and these are things that molecular biologists work with every day. They're binding and unbinding systems. They're diffusive systems. They're systems that are not driven by any energy forces, or any chemical potential. So, for example, if you transcription and translation are not detailed balanced because ATP is driving those systems as a fuel molecule. But in many systems, again, I always say binding and unbinding reactions, when things bind together and then unbinds, are detailed balanced. They're very common. They underlie many components that are driven as well. And what we've shown is that this large class of biochemical networks is deeply similar to a large class of machine learning algorithms of generative models. And by sort of taking this math, in effect, and saying we can use ideas of inference and generative models for machine learning to understand what's happening in biochemistry. And we can even take that a step further and ask, can biochemistry learn the way a machine learning model does? And the answer is yes, it can. And we can build models of biochemical systems that are not particularly complicated. They're things you could implement in a lab, and certainly things that could evolve that learn and adapt in a way that is exactly the way machine learning algorithms, or some types of machine learning algorithms, learn and adapt.
What would you like to see come out of this? Like, do you think this is a good way to do a neural network, or is it kind of more broad than that? What kind of systems might we use this kind of framework for?
It's not a neural network. It's different. It's a generative model. So it's, if you're familiar with graphical models or Boltzmann machines, that's really the model. So it's a probabilistic model that can generate and respond to the environment. I think that, these models are very powerful. They're hard to train in silico, but they're also models that, a lot of neuroscientists think that generative models are sort of underlying many functions of the brain. So I think that thinking about these models as a way biochemistry or biology in general computes will be very fruitful for understanding how things work in the real world. Certainly, if we could implement these models in silico, that would be cool. And one possible thing that this paper we're working on might do is help people invent new machine learning algorithms in silico. I think it might also help us interpret existing biochemical motifs and functionality, not as a program or not as a conventional logic program, but instead as a sort of machine learning generative model program. And if we start thinking about the way biochemistry functions as a generative model, as a machine learning algorithm, I think that might be fruitful for understanding how it works, why it works, how it breaks, and how to fix it.
So how far do you think the theory work you have been working on and you plan to work on far from the real-world implementation? Do you think it's very close, or there are some other obstacles on the way?
We could go into the lab right now and build simple versions of the theory I'm working on. The problem is that they wouldn't be very interesting. They wouldn't be interesting because what the theory really relies upon is small molecular counts. So, and I think this is actually kind of deep, but and not a proof. I'm waving my hands here a little bit. But we have some strong anecdotal evidence that somehow by having low copy number, like 1 or 2 molecules in a volume, you can actually do more sophisticated computations than if you have 100,000 molecules in a volume, or Avogadro's number of molecules. And I think this is really interesting because it sort of, tells you a good place to go experimentally. We should be working with small volumes and low copy number. That's hard right now. People can do it, but it's challenging. But it also tells something about biology. It's always been amazing to me that there's 1 copy of your genome or 2 copies in every single cell. Why 2? Why not 10? Right? Well, maybe there's some deep physics going on that actually makes 2 a better number than 10. And I can't prove that right now, but it's definitely a hypothesis and a conjecture that I think would be really worth exploring.
Yeah, I think that would be interesting to explore in the context of I think the Lakoff-Ron is a classic example of where you get maybe on the order of 10 molecules of maybe Georgios, do you remember?
I'm not sure on the exact copy number, but there are tons of bacterial systems which exploit low 1 to 2 or sub single molecule level numbers just to perform. But I wanted to ask, can you give any intuition behind why the low copy number, why a low copy number system might be able to do more complicated computation?
Yeah, actually, here's one using the lac operon. So if you look at the lac operon, it has 3 lac repressor binding sites. And so the lac repressor tetramer can bind to any of those 3 sites. And more importantly is that you can have the lac tetramer can cause looping between the DNA. So if you have 3 sites, there are 3 possible loops you can form. And 2 of these 3 loops repress transcription really strongly. Now let's imagine that I have 100 copies of this gene, of this operon, and I can form these 3 different loops or no loops. Well, statistically, I'm going to have a mixture of things where some things are looped in one conformation 1, some are in conformation 2, some are in conformation 3, and some are unlooped. And that means I'm going to get a mixture of transcribable genes and untranscribable genes. While if I only have 1 copy, I can really say, that 1 copy, or if I have 2 copies, they're, 2 out of the 3 states are likely to be in the turned off loop. And that will somehow more tightly regulate my system. In the world of physics, this is really an entropic effect. We're seeing that when you have sort of combinatoric sets of states, entropy is going to drive you to explore some of those combinations, but you really might want to restrict the combinations you can explore at any given time. And by restricting the state space you can explore, restricting the possibilities, you can restrict the functions that will occur. And that makes your computations more powerful. Because not everything is happening all the time. Some things happen conditioned upon some events.
How do you think this might interact? So you mentioned, for example, low copy number of genomes, but there are some scenarios where you have larger copy numbers, like plasmids in bacteria, or when mitochondria fuse into networks, and plants. Do you think this idea might be able to explain that as well?
Well, I mean, mitochondria do fuse into networks, but typically there's one small volume with one mitochondria genome, or maybe a couple. I think it's more interesting to ask, there's lots of bacteria out there, but no one's ever seen a bacteria that only has plasmids. Why is that? Well, to me, that is very suggestive that maybe there's something not as good about plasmids. Plasmids are great for sharing information between bacteria species, so there might be some pressure to have some plasmids. But underlying the whole diversity of bacteria out there, or viruses, I've never heard of a multi-genomic organism. That's not completely true. There are some plants that are sort of like giant single cells, some protists that have many genomes, but they're spatially isolated. Each copy of that genome is going to be fairly far away from other ones. So I've never heard of like a stack of genomes that are all co-inhabitating the same space. I would love to hear about it. That would be a fascinating life form.
There's one thing I have heard of, but I don't think it quite meets that definition, which is trypanosomes have, I believe it's called a kinetoplast, which is this kind of chainmail genome sort of thing. I think it is the mitochondrial genome, and it's split into a bunch of these rings which are catenated together in some complicated way, but it's not many copies of the same thing, so I don't think it is an exception to what you're talking about.
Well, and again, if you have some things that are higher copy number, that might be okay. So like plasmids are not really doing a lot of sophisticated circuitry in bacteria, except when synthetic biologists go, build logic circuits on plasmids, which don't work as well as genomically integrated ones. Typically what plasmids are doing is they're just providing some protein of interest. And that's very different from, say, the logic underlying how you run a bacterial system, which seems to be genomically integrated for the most part.
It's kind of a different question. When you went to start your degree in biological physics, or maybe before, had you heard of molecular programming? When did you, or synthetic biology? When did this become kind of the thing you wanted to do?
That's a great question. So when I started my undergraduate degree, I was interested sort of in physics. And I liked biology, and there was like a biological physics freshman seminar, which I took, and I thought was cool because the math was hard, but it wasn't about like quantum particle physics that really have no bearing to my life. It's like, this is how things in my body function, and here's some really hard math I could do. I love doing hard math. And I kind of kept going down that track and just saying that, like, oh, there's so much complexity here and so many unexplored questions, and they're different and distinct from what sort of other people in physics were doing. And honestly, they required just as much, they were just as challenging in many ways, but weren't as headache inducing to me. Like, I was always like fed up with electrons in boxes. Like, I don't care about electrons in boxes. There are no electrons in boxes, really. I was much more interested in like protein folding. That's like, yeah, there's lots of proteins and they're all folding. That's fascinating and way harder in some ways. So I sort of kept getting more and more drawn into biology, and I realized that computers were part of the way we could solve these problems. When you're dealing with complexity, you could write down the physics, the laws of physics that explain biology are fairly well understood, but everything happens when you mix and match them in complicated systems, and that requires computers to really handle. So, my undergrad, I did some polymer physics and some simulations and experiments around sort of single strands of DNA and what they might do when you push and poke them in different ways. Then I went on to say, I really want to understand networks. And it's actually a professor, Gary Wessel, who inspired me to the notion of synthetic biology. He had a class at Brown that I took, which was fantastic. It was really just sort of a lit review course, but it was opened my eyes to sort of, we can build cool things. And on the other hand, another professor, Tom Powers at Brown, recommended I read Yuri Alon's book on, design principles of biology or something like that, on circuits. Great book. I'm butchering the title, but and I read that and I was like, we can use, math to understand these circuits, and then we can program them. And it seems like very, very cool. I went on to do some bioinformatics after graduating because I wasn't sure if I wanted to go to grad school or what. I knew a lot of statistics and coding. I was at Institute for Systems Biology doing cancer research for a few years. And I really saw that, the big data approach we're not ready for it because we have too many holes in our mechanistic understanding. And so then when I was looking at grad schools, I was really trying to find a sweet spot in the middle of sort of this big data systems biology, bioinformatics world, and the sort of molecular biophysics. And that's what led me to molecular programming, which sort of sits right in the middle. And I'm very much drawn to, still getting to use all the physics I like and learned, still getting to write code and solve hard math problems, but also, help build things that exist.
And do you think, back when you were, say, 10 or 12, would it be a surprise to young William to see where you are now? Like, would you be at that age just thinking, I'm going to be a physicist, or what was your view on your future then?
I mean, I love Jurassic Park, so I think I would have been pretty down to doing some serious molecular engineering and building some dinosaurs, or very small dinosaurs, the case may be.
Okay, so we've covered your theoretical and computational work. But you've mentioned a couple times your interest in E. coli or cell-free extracts of E. coli. Could you tell us a bit about what your interest in that space is?
Yeah, so I mean, as I mentioned, I think a lot of it's sort of practical. I think that doing theory and computation without any notion of how a wet lab works is detrimental to my success. So cell extracts are nice because you can do sort of high throughput experiments, and they don't do things like die on you or evolve away your genes that you put into them because they're not alive. So that makes them sort of a stripped down version. At the same time, they're more complicated than the DNA nanotech you guys hear a lot about on this podcast, I'm sure, because they're closer to being alive. They once were alive. It's sort of, someone once described cell extracts to me as necromancy, and I kind of love that because we're taking dead things and we're reanimating it in order to understand the living, or to engineer the living, which is a little bit of an odd perspective. But fundamentally, I don't think cell extracts are necessarily that useful. I think they're a great platform to test ideas on, sort of like a hydrogen atom, but less so. I also think that, they they sort of explain a lot of the problems we have in biology, which is complexity, dealing with unknowns. And they've got to me at least interested in metabolism, which I think is one of the things that differentiates biological computation from other forms of computation, is that biological computation can interact with matter. It can produce things and degrade things. And I see it as a technology not really taking over what computers are doing, but rather taking over what static objects in our lives are doing and imbibing them with computational powers and regenerative powers. And so to me, that's really interesting. And you have to start with the basics of like, how does metabolism work? How can you produce and degrade things? How does fuel flow through living systems? And we barely understand that with cell extracts. So I think that trying to do that at a higher scale is going to be harder until we can do it at like the simplest possible, not even a live scale.
So I think kind of the more short or medium term application you're kind of hinting at is with the use of metabolic engineering is this notion of green economy. But I want to start with what you're talking about of imbuing static matter with the ability to think and compute. What's your long-term vision for what this kind of technology and these ideas will bring to the world?
Well, I can put my nerd hat back on and talk about elves and their tree houses and say that it would be really great if we could plant a seed and it would grow into a living house for us. And I don't think that's actually impossible. I think that is scientifically a reasonable dream to have in like a couple centuries. And so, what does it take to lay the foundation for that? Well, we really have to understand how to program from a simple set of genetic instructions, geometry and the metabolic or energy mechanisms inside whatever thing we're trying to build is. And that's molecular programming. So my dream is sort of to couple all of these areas.
And what would, if you could have one thing from, say, 1,000 years down the line, whatever molecular programming brings us, whether it be tree houses or regenerative health or anything, what's the most imaginative and thing that you can think of that you'd want to have right now?
That I'd want to have right now? I mean, I'm really, as you hinted at, I'm really concerned about climate change. And so I think that if we could have living materials that sequestered carbon for all of our materials that we use for everything from concrete to steel to houses to wood, which was all alive and all growing and self-sustaining and sequestering carbon, that would be fantastic. And, I don't think it's far-fetched in the long term, like, thousands of years, that we will get there, that we can learn how to live, engineer, and cohabitate this planet by using living organisms to build a habitat for our species that is not destructive, right? But that requires us to not just mesh our lives with living organisms the way, by having preserves of, wilderness and then human cities. We have to go a step further and enmesh life into our very infrastructure.
I guess you'd view it as re-terraforming the Earth.
Yeah, exactly.
So how might we get to that point? What's the first step in building this future?
I mean, there's lots of first steps. I think there's no shortage of first steps. One of them is understanding how to program with biomolecules. Another one is understanding how developmental biology and how pattern formation can work from a single seed. A third is understanding, how to program the metabolism and energy so that the products we need as humans can be produced from non-petroleum sources. There's endless possibilities of first steps we can take.
So, you're co-supervised by Erik Winfree and Richard Murray, two quite titanic individuals, one in kind of the molecular programming space and the other in control theory and synthetic biology. What's it like being supervised by these kinds of people? Do you get much face time, or do you kind of interact more with their postdocs and other graduate students? Yeah, and I suppose the working in kind of these large groups how has that changed your graduate school experience?
Well, being co-advised means you get twice as many meetings, so that definitely has its disadvantages. My projects are fairly influenced by each other, but I work on separate projects with each faculty. Erik has been a fantastic advisor in terms of hammering theory into me in a way of doing it more rigorously, which is great. He also runs a very small group. Right now, I think there are 2 graduate students in his group and no postdocs. In the past, it was as big as maybe 5 or 6 people, but he works more broadly in a group of other researchers at Caltech, including Lulu and Paul Rothman and Lulu Chen. And so that gives sort of a bigger group there in general. And what I like about that sort of supergroup, they call it the DNA supergroup, is that there are experimentalists and theorists from very diverse backgrounds, ranging from biochemists to computer scientists to renegade physicists like me, who are all working together and learning to speak each other's languages. And I think that is very, very fruitful for collaboration and understanding what other people need and making your research more accessible. Richard's lab is, I think, a little bit more of a traditional lab with quite a few postdocs, quite a few grad students, mostly bioengineers, but occasionally there'll be a control theorist or someone like me coming in from the side. And I also really like that I get to work with more traditional bioengineers doing more traditional work. I think that's super important. And Richard and Erik have totally different styles. Erik will sit down with you in a room for 4 hours. We just talked yesterday about this paper for a very long time, and we hammer out math. Richard will not do that. He's way too busy. I hear he has robot versions of himself. That way he can take care of all of his commitments, because it's amazing what all the things he does. But he also gives, he will give you feedback on every single paper you want to publish within a couple of days. It's fantastic. But it won't be the type of I'm going to help you hammer out your math feedback. It's going to be much more practical feedback about here are things that, will make this more understandable to someone who doesn't have your math background. You should work on that.
How did you end up with 2 supervisors? Was it a case of you couldn't pick between 2 fields? Or?
Well, I wanted to do multiple things. So when I was talking to Erik about joining his lab, he wanted me to do certain experiments. And I was not terribly interested in doing DNA nanotech experiments. No offense, you guys, but I find them sometimes a little boring. It's not really, I found them more interesting as I've gotten to understand the nuances better, and I understand why people do them now. But as a first-year grad student, I was not I was wanting to be closer to biology. Same time I was talking to Richard, and he was about to go on a sabbatical, and he was not willing to take new grad students unless they were co-advised. And I didn't really want to join Erik's lab without having closer proximity to experimentalists who could help train me to do the experiments I wanted to do, which Richard's lab provided. So it worked out very nicely that I could both get the experimental support I wanted. At the time, there was one experimental postdoc in Erik's lab, Chris Stachuk, who's now at the UW, really talented, but I was like, he's a postdoc, he's going to be leaving within a year. That was wrong. He spent a little bit more time than that, but I didn't want to wind up being the only experimentalist in that group. And Richard didn't want to take me if I wasn't co-advised, so it worked out very well.
So going back to the synthetic biology applications and the green economy and adding lots of carbon sequestration into the products around us and the way we manufacture, I guess this starts to bring into more focus the idea of regulation, biosafety, bioethics, which I think the field of synthetic biology has taken a strong stance on from early on. I think that it's something which they focus on a lot. But do you think we're ready for these widespread applications? Do you think there are any concerns we should have, or should we go as soon as possible to trying to fix climate change with synthetic biology?
Well, I don't think we can fix climate change with synthetic biology very soon, so I think we have time to work on these answers. I do not think we should dodge the regulation and ethics bullet by just saying climate change is more important. I think we should absolutely be having these conversations regularly and consistently and very carefully. That said, I also think there's lots of inefficiencies in the system. So right now, at least in the United States, there's 3 different government organizations that claim control over GMOs in different contexts. and there's no legislation about how GMOs are regulated from the federal government. That's a problem because that means there's no consistency and you can't bring new technologies to market, which is what we need. So that has to be somewhere between, the laissez-faire world that is most of tech and the incredibly tightly controlled world that is medicine. I think synthetic biology, depending upon the application, will be somewhere in that range, but presumably much more controlled than tech. But if we're not eating it, if we're not inducing it and putting it into the wild or ourselves, less controlled than medicine. And right now, it's probably neither here nor there.
I think, was it briefly mentioned that you're nearing the end of your PhD?
Hopefully.
What's next for you?
Good question. I ask myself that. I'm talking to companies, I'm talking to professors. Some of it will depend upon where my wife wants to go with me.
You said you're talking to companies, so you're not committed to academia.
Not necessarily. I think that part of the problem is that for what I want to do, we need a lot of data. And, you mentioned what my wet lab work is. Part of my wet lab work was trying to collect a data set big enough to use for ML. Didn't work. I spent $20,000. Was not big enough, not even close. And, and this was the simplest data I could imagine, metabolomics on cell extracts. Who has the resources to really collect huge amounts of data that we can actually try to do things? It's mostly industry. So I'm definitely interested in having access to that type of data. And that probably will require industry connections in the long run. I am perhaps, that's the word I'm looking for. I'm optimistic, but perhaps unfoundedly so, that it's possible to bridge academic and private sector to some extent back and forth if you play your cards right. And you definitely see this happening in the computer science world, a little bit in bioengineering and synthetic biology, but again, these areas are newer, so it's unclear how well that will work. But that is sort of, I think, the track I would like to be on, because scale comes from private sector and government, but mostly private sector. But the real research is still happening in academia for now.
Yeah, I want to take back my shock at at you considering leaving academia. I think that needs to be. I think that's something that needs to be normalized. That there is good, that you can do good research outside of academia. And I think you just highlighted one of those things is industry can have way more resources than the average academic lab to undertake massive projects. And so you said you're considering industry. Do you also consider maybe the startup route, doing everything, just do it yourself, get the funding yourself?
Well, the companies I would join are mostly going to be startup type companies, but I don't want to be a fundraiser CEO. I want to do the science. So at least at this point in my career, I'm not very interested in being a founder. That sounds like a lot of schmoozing. I want to do math and not talk to people. But to your point about sort of scale, I think this is something that has bothered me a lot about synthetic biology and biology in general, is that most labs work as these isolated little kingdoms with their, few $100,000, maybe $1 million if it's a well-funded lab, up to a dozen or 2 dozen people, and they do what they can. And we're hitting a barrier, I think, in what we can do. And you look at, grad students will now spend 6 years on one little circuit that is the next, one step forward for synthetic biology, or 6 years on the next piece of, a DNA circuit that's less leaky or 10 times bigger than the last one. And then it's never really reused again exactly. We really need to start a collaborative type of academic endeavor to really engineer biochemical systems that will require working in much larger groups, and in a totally different way, and using much more money in a collaborative way. And I think physicists have this down with things like the LHC, where they come together and spend billions of dollars and accomplish incredible things and all benefit from it. We need to do that in synthetic biology and systems biology to really move to the next level in academia. And I would love to see academics come together and form giant collaborations and get $100 million to do something out of this world. That would be cool. Maybe we could build a cell. We could. And one of the exercises that Bill Cell was how much will it cost and how long will it take? And I think we said, $1 to $10 billion over 10 years and we could do it. But that type of money is not flowing in single pots. It's going to lots of little pots, and there's no centralization to really make this happen as a community. And when we do centralize, the academic credit is all about the first and last authors on these papers. So a whole bunch of people in the middle are screwed. And that needs to change. We need to change how we give credit so we can work in big groups and accomplish bigger things.
I think we're seeing things like that begin to emerge. For instance, in London and Edinburgh, there are these DNA biofoundries where you can, at a huge scale, well, relatively large scale, synthesize loads of unique genetic circuits and screen them. I wonder if there's anything like that in the US, but I wonder, what do you think needs to change in academia for us to kind of come together and do these international multi-billion-dollar LHC equivalent projects? How do we convince academics and also funding bodies?
I mean, I don't know how to convince them, or else I would be out trying to do that and getting the money, because that would be cool. I think funding bodies, if academics said we want to do it this way, they would give the money that way. The funding bodies usually listen to the academics pretty closely. It's about getting academics to agree to it and to agree to working together and to breaking their traditions around some 10-year traditions and other traditions around authorship. In the US and the NIH, we've done some work like this. The Cancer Genome Atlas, where I did bioinformatics, is a $100 million, $200 million collaboration between many hospitals and labs and data centers. As an example, I think of at least an organizational success story. I'm not sure if they learned a lot from that, but certainly it was a lot of people and a lot of money working together. The biofoundries I know many people at the biofoundry at Imperial, Paul Fremont's group in particular, and that's not even the scale. There you might have a little bit more money and some more robots, but there's not technicians operating those robots for you. You still have to learn, if you're a grad student, how to use all the robots yourself. It's not really the type of collaboration that we need, where we have I can come in as someone who can help build models really, really quickly and well, and then work with someone who can do cloning with a robot who's super specialized in that, and then build 100,000 constructs because we have everything's all there for us. It's not really that easy, and there's not money for it, because that even would cost hundreds of thousands of dollars of reagents and things to make that happen, and time, getting lab techs and etc. And that money doesn't exist, even though there's some robots.
Do you think there's like a cultural tipping point where you can envision a certain point at which the field would transition to being able to do these large projects? Like, what is it about the field of physics that allowed things like the LHC to come about that isn't currently a property of the biological community?
I was just asking my great uncle about this, who is a physicist. He, is 80 something now. His work is on neutrinos, so he was part of the neutrino detectors in Japan under the mines. These are $100 million collaborations across huge international groups. And he said the reason why physics does this is because they all learned how to do it working on the Manhattan Project during World War II. And he was trained by someone who worked on the Manhattan Project, and all of his contemporaries were trained by people who worked in this giant scale run by the government, I guess almost 100 years ago now. 90 years ago. And that's how they learned to do this. And after that happened, they had this success story that if we come together and work, we can do world-shattering experiments. And they took that and kept running with it. That's never really happened in biology. But it should for climate change.
The Human Genome Project?
The Human Genome Project. Yeah, I mean, I think that the ISB where I worked was born out of the Human Genome Project as an example in Seattle. It's run by Lee Hood, who was one of the main players in that. And they're trying, but again, it's the majority of biologists and aren't on board with that, right? They're working in their own little labs. And it would require a cultural shift where the majority of biologists would have to start working in these collaborations for the funding industries and for the tenure, committees of universities to really change how they evaluate these things. And until that happens, if I can be middle author on 100 papers, I'm not going anywhere in biology or bioengineering. In physics, that might take you very, very far if they're the right papers. And if you're known in the field for being a good guy or girl who can do great work.
So what's your dream biological equivalence to the LHC or the space station? Let's say you had the funding and the institutions, and they gave you several $1 billion and tens of thousands of researchers. What's our best use of that?
I would build a synthetic cell is the first thing I would start with. I would do it in a way where we know everything that's happening, have models of it, can improve upon it, reverse engineer it, re-engineer it, make it modular, and then I would take that and make that into a synthetic multicellular something. And I would just go from there. And I think once we have that foundation of one cell, from there, all of life evolved. It will not be hard to engineer that, but we have to start with something that we really understand in order to add more and more complexity.
Is there an argument to be made that in biology, and this might be controversial, but that in biology, the breadth of questions you can ask and the breadth of research you can do is maybe larger than in physics? Like, okay, ignoring kind of macroscopic physics, if you're interested in fundamental physics, there's, I would say, a narrower set of questions you can ask. Like, if you're interested in particle physics, then you're trying to look for exceptions to the standard model. Is probably the most likely thing you want to do. Or if you're interested in cosmology, you might be looking at kind of ground unification theories. I feel like in biology, there's just so many more questions you can ask that is it harder to get the entire community or even a large subset of that community to come together for one project when they might have their own ideas of what they want to do?
I think a lot of biologists would agree that there are many questions you can ask and even more ways you can answer them experimentally as part of the problem. Like the LHC is the only, particle accelerators are the main tool that we have for theoretical particle physics because we need high energies, and that's how we can get them. But at the same time, like if you look at all of the cool quantum stuff, quantum topological phases and that crazy stuff, that happens on the benchtop. And some really cool physics is happening that I think is honestly probably going to be more useful in the short term than anything we're doing with particle physics. I don't think that it's the questions. I really think it's a mentality thing. And if you look at something like the LHC or LIGO, they have to invent new devices, and the solid state physics people are involved in the devicing for these things and the detectors. When you actually go towards a really lofty goal, it's going to wind up sweeping the whole field with it, provided that the goal is hard enough. If the goal is really leaving out a lot of people, that probably means your goal is not lofty enough.
So you have mentioned that you like to consider industry because of the money and scale and maybe credit. And other than this, is there any other reasons you would prefer industry, or do you see any other difference for a person who do not know the difference between industry and academia?
I don't know the difference. I've never been in industry. I think that for me, the most important thing about what I do personally is that I'm working with a bunch of smart people doing cool stuff. And I care about the types of projects I work on and what I'm doing day to day a lot. So, even if you pay me really, really well in industry, if I end up doing a bunch of work that makes me miserable, I'm not going to have a lot of fun. Same with academia. Even if I have the most cushy tenure track or tenure professorship, but I have to do a bunch of work that I don't find interesting, I don't want to be doing that. Or working with people I don't like. So I'd rather work with a group of people I really like on a project that I find challenging and rewarding than anything else. And that could be industry or academia. And I think it's all about the people.
Thank you so much for joining us, William. Stay tuned to our newsletter for details on our next podcast episodes. Thanks for listening.